Hi, my name is Aiyana Houston. I've recently graduated from Shawnee Community College and I'm currently a zoology major at SAU. So the research project I've worked on is the effects of Mesophication on genetic diversity, epiphytic bryophytes on oak versus maple trees. What exactly is a bryophyte? I'm sure you've seen these small green plants growing on rocks, on trees, or even on the side of your house. Those are bryophytes. Bryophytes are small flowerless green plants that do not have roots or vascular tissue. Bryophytes are epiphytic, meaning that they grow on living plants for physical support, such as oak and maple trees. We specifically wanted to look at oak and maple trees because of a process called mesophication. Mesophication is when you remove fire from a fire-dependent ecosystem, and it completely changes the whole structure of that ecosystem. This is currently happening in southern Illinois with oak and maple trees. So we wanted to assess the biodiversity of epiphytic bryophytes on oak and maple trees by identifying whether they were generalists, meaning that they do not have a preference between oak and maple, or they're specialists, meaning that they do have a preference between oak and maple trees. We did this through morphology and DNA barcoding. The three DNA barcoding regions I looked at were RBCL, tolerant, and RPS4. So the identification of epiphytic bryophytes will help us understand the relationship between the substrate and evaluate how processes like mesophication can impact bryophyte biodiversity. So we had 28 samples were collected. 14 were from oak trees and other 14 were from maple trees. I then did microscopy. I took images of branch and stem leaves at different ramifications. I then took measurements of those leaves, and then I took the images and measurements then compared to the dichotomous key, which can be found at Flores in North America. So I then did DNA barcoding. I took the genomic DNA from 13 of my samples, and I ran PCR, and then I did agarose gel electrophoresis. So these are my results. I put my results up into the two different approaches I've used for identification. So for identification through morphology, what I did was that I took the characteristics I noticed in my sample and compared those back to the dichotomous key. For example, you could tell for sample 20, my apex here is acute, and that was a characteristic of Lutzkia polycarpa. I did the same thing for my sample 6. You can see the cells here are very different, and that's my alar region, and that is a characteristic of Intodontidatrix. These are my identifications of DNA barcoding. These are the images of my gel electrophoresis. This is my RPCL gene at approximately 600 BB band size. This is my turn lift gene, and it's approximately 400 BB band size, and this is mine. Our PS4 gene is approximately 600 BP band size. So we identified 13 epiphytic bryophytes, and they were confirmed by comparing the samples to that of the dichotomous key. We did find some generalist species, those being Litzkia graciliscens and Dodon coloricens. Those were found both in oak and maple trees. But we did find some specialists. Those were Litzkia australis, Litzkia polycarpa, and Intodontidatrix, and Intodontidae, and those were found only in oak trees. So the analysis of my gel electrophoresis confirmed the presence of all the three genes I was looking for. So in the future, I plan to continue identifying my remaining samples through morphology and then do DNA barcoding. I then would like to make a phylogenetic tree to evaluate the differences in bryophyte biodiversities at oak versus maple trees. I would like to thank Southern Illinois University, School of Biological Sciences, the SI Bridges Program, NIH, for the research opportunity and funding. I'd also like to thank my mentors, Dr. Laxmi and Joel Green, for helping me throughout this project. I'd also like to thank the Noise MTF program for collecting the bryophyte samples.